The overall goal of this procedure is to demonstrate an atmospheric pressure plasma setup that excludes air from the reaction environment.
Our setup helps answer key questions in the field of biomedical non-thermal plasma, such as the origin of reactive species that define the efficacy of plasma treatments of biological samples. The main advantage of the setup is that it excludes the uncontrolled ambient environment. The atmosphere inside the reactor consists only of the feed gas components and the evaporated sample. The significance of this technique extends toward clinical applications of cold plasma, because it provides fundamental understanding of physicochemical processes that take place when low-temperature plasma interacts with liquids.
This method not only provides an insight into the effects of the biomedical plasma, it can also be used for other purposes, for instance studying chemical reactions in a controlled environment.
Demonstrating the procedure will be Philip Groves, a postgraduate student from our laboratory.
First, design and build a Faraday cage from wire mesh to shield the equipment. The cage must be large enough that the live electrode, the ground electrode, and the electrode cables will not contact each other or the wire mesh when all equipment is in place.
Plasma jets operated at kilohertz frequencies are extremely susceptible to external fields. The Faraday cage creates a stable electromagnetic environment, eliminating the influence of the surrounding equipment on plasma and protecting the operator and equipment from plasma-generated electromagnetic fields.
Ground the cage and all metal supports inside the cage using a device appropriate for the electrical socket type. Affix a warning placard to the cage. Connect a helium gas cylinder to two mass flow controllers.